Here's a question for you. If you cannot see something, does it mean that it is not actually there? Do you believe in the supernatural? Do you believe in the existence of ghosts? Perhaps you do, perhaps you don't. Maybe you're not sure. At one point, the scientific world and medical world were not sure of the existence of bacteria and viruses. And they would carry out operations in environments that were far from sterile. Bacteria are tiny organisms with flagella, plasmids, nucleoids, and they cause infection in some cases. The only reason we know of their existence as entities that cause or potentially cause harm is through advances in science and equipment that has helped us to see them and study them. Viruses are invisible to the naked eye, but science has also created instruments that allow us to see these tiny subcellular parasites. And so one of the major reasons why we study cells, animal cells, plant cells, is because we want to understand how viruses and bacteria cause damage or pathology. Pathology to us, pathology to animals that we farm, and crops that we farm. And as part of both the T-level and A-level syllabus, you have to know about the various different structures within an animal cell and how they produce proteins, lipids, energy. So think about the different structures that you see before you right now. Think about what they do. What's the function of the nucleus? What is the function of the nucleolus? What does a ribosome do? Mitochondria, the Golgi apparatus, rough and smooth endoplasmic reticulum, centrioles, cytoplasm, and the cell membrane. In fact, there are many membranes throughout the cell that are composed of a phospholipid bilayer. And that is something that you don't get in prokaryotes or bacteria. There are many different types of cells, and cells are specialized according to what activities they do within the human body. Red blood cells, for instance, do not have a nucleus in order to be able to carry as much oxygen as possible from one part of the body to the other. Nerve cells are specialized. They're long and they're branched. And it's my suggestion to you to learn how to label some specialized cells that you find in your textbook, or perhaps label up a nerve cell, a muscle cell, and a skin cell. and just become familiar with the different components within and how they relate back to the basic structure we saw on one of the previous slides. You need to know how to label up a bacterial cell and where its cell wall, cytoplasm, plasmids, nucleoid or circular strand of DNA are, as well as the fact that some bacteria have flagella and some don't. Some have shorter projections or a singular short projection called a pilus, which is involved in the transmission of plasmids and disease resistance from one bacterium to another. So there is a hidden world all around us and a hidden world within us. The hidden world comprises bacteria, viruses, fungi, fungal spores, and even the bacteria that live and exist within our gut. Scientists use many different methods to cultivate and grow bacteria in laboratories. Look up streak plating and how bacterial colonies can be cultivated on different types of media, including blood agar and even chocolate agar. So viruses, bacteria, fungi, fungal spores exist all around us, and some of them are dangerous or pathogenic. And so we need to take precautions whilst working with them. Now here's an interesting plate. The small yellow discs are small bits of filter paper soaked in various different types of antibiotic. And this method is used to determine whether an antibiotic is effective or not and able to kill a particular type of pathogenic bacteria. So the take home is it's very important to sanitize both in the lab, in healthcare, and generally, because there are bacteria, viruses, fungi all around us, and some of them can cause disease. And they live within us and throughout the entire planet. So, though we cannot see bacteria, fungi, viruses, they exist, and so too does the potential to cause us great harm.